The gut contains hundreds of different types of microbes that are highly person-specific. It has been challenging for researchers and clinicians to identify and pinpoint strains that aid in the interaction between the gut and the immune system. Methods have included fecal transplantation as well as monocolonization, colonizing animals with a single bacterial strain to discern specific bacterial mechanisms of action. While the latter approach has identified bacterial strains capable of modulating immune cells, it does not consider that a strain exerts different functions when in concert with other bacterial species in a community. Recently, synthetic models of a human gut community have been created to study the functions of the gut microbiome in vivo. One recent finding indicates that instead of one T cell recognizing one particular microbe, a small group of T cells recognize a broadly conserved antigen shared among many bacterial strains. This discovery opens a therapeutic opportunity to skew gut immune reaction towards tolerance or inflammation by manipulating microbiome-specific T cell response. Without further ado, I'm very pleased to introduce our first speaker to you now. Dr. Nagashima received an MD and a PhD from the University of Tokyo in Japan. After completing his postdoctoral training at Stanford University in California, he will start his laboratory August 2024 in the Department of Molecular and Cellular Biology at Harvard University, Cambridge, Massachusetts. His research program uncovers human immunology, microbiome, and diet at the level of molecular mechanisms. Dr. Nagashima is the winner of this year's annual Noster and Science Microbiome Prize. We included his prize-winning essay, Blockbuster T Cells in the Gut, as well as more information about the prize in the additional links. Welcome, Dr. Nagashima, and thank you for joining us today.
Thank you so much for your kind introduction, Jackie. Can you hear me?
Yes, I can.
Okay. Hello, everyone. I'm Kazuki Nagashima, a senior research scientist of Michael Fischbach Lab at Stanford University. It's my pleasure to give a talk here. I'm very excited to talk about our recent publication. In this project, we try to understand the complex interactions between the microbiome and T cells. We map strains of the gut microbiome to the T cell receptor repertoire. To study the gut microbiome, people have relied on two experimental formats. First, we often transplant a fecal sample, an undefined bacterial community, to germ-free mice. Many papers found impressive phenotypes by fecal transplantation. Examples are anti-PD-1 therapy response in cancer, viral response, and obesity. But with this method, it's hard to identify the mechanism. Another approach is that people monocolonize a single organism to germ-free mice. An interesting phenotype is observed. However, the phenotype of a single strain is different in a monocolonization and in the physiological background of other members of the gut microbiome. So we all sought a system with a physiological complexity of fecal samples, but with the defined nature of this experiment, so that we could manipulate the bacterial community afterward to test the mechanisms. To make this happen, we built a defined community with a large number of strains from the human gut microbiome. Based on our pioneered work by Alice Chen, a talented postdoc of Fischbach Lab, we built this complex defined community HCOM, a collection of 104 bacterial strains. We wanted to make a model system of the human gut microbiome, so we reanalyzed the NIH Human Microbiome Project data, and we selected the highly prevalent strains, the strains that reside in almost every human gut. Since HCOM is composed of the strains that every human has, HCOM works as a standard model of the human gut microbiome. A powerful and convincing approach in biology is to delete one factor from the entire complex system. If we delete a gene from the genome, we can examine the function of a single gene in a complex organism like a mouse. However, we cannot delete a specific strain or gene from the gut microbiome. We have relied on simpler methods, including monocolonization. But monocolonization is very artificial, so we seek a way to delete one strain from the gut microbiome. HCOM, a collection of 104 bacteria strains, makes this reductionist approach possible in the microbiome field. Each strain from a library is kept as frozen stocks. We revive each strain separately in a 96-well plate. Each strain is normalized by OD and pooled to inoculate germ-free mice. If you want to test the function of one strain, you can simply avoid mixing the strain before colonization to germ-free mice. Also, every single strain is defined, so we can test the function of each strain separately in this setting. We tested the stability of HCOM when colonized into germ-free mice. Each colored dot indicates one bacteria strain, and lines connect the same strain. We analyzed the relative abundance of each strain by metagenomic analysis in the inoculum and in the fecal pellets every week after colonization to germ-free mice. Strains are very stable after 2 weeks of colonization, even though inoculums are slightly different run to run. The composition of the community in vivo is nearly identical every time. From now, we analyze mice after 2 weeks of colonization when the community structure is stable. We first examined what immune cell subsets are induced by HCOM colonization. We revived HCOM strains and pooled them to inoculate germ-free mice. After 2 weeks, immune cells are extracted from the large intestine and tested by FACS. We tested every immune cell subset, but the biggest effect by HCOM was observed in T cells. IL-10 positive Treg cells and IL-17 positive Th17 cells were tested. The T cell profile of HCOM colonized mice was similar to that of SPF mice and distinct from germ-free mice. HCOM doesn't have SFB, a strong Th17 inducer, but shows a physiological level of an increase in Th17 cells. We conclude that HCOM colonized mice work as a model to study immune modulation by the gut microbiome. Here is the question we address today. Previous works from Dan Littman's group showed that SFB, segmented filamentous bacteria, induces Th17 cells that is antigen specific to SFB. Helicobacter hepaticus induces Treg cells specific to Helicobacter hepaticus. However, it is unknown what interactions occur between a complex community of the gut microbiome and individual T cells. To answer this question and map T cell specificity against each strain from HCOM, we established a new assay. We revived HCOM strains. HCOM strains are mixed and orally inoculated into germ-free mice. We waited for 2 weeks to give naive T cells time to differentiate, then isolated T cells from the large intestine. We co-cultured gut T cells with each of HCOM strains, one at a time, using dendritic cells for antigen presentation. So we have 104 conditions since we have 104 different strains. We used FACS to analyze T cell receptor downstream molecule NR77 expression as a readout for T cell receptor stimulation. By looking at NR77 expression in T cells, we can tell which strain stimulates T cells. This is the result of the co-culture experiment. I begin with one example shown in the right side of the panel. I gated on P-Treg cells and examined percentage of NR77 positive fraction by FACS. When P-Treg cells are co-cultured with Intestinobacter barzillettii, the average percentage of NR77 was 10%. When compared to a PBS negative control, p-value was 0.01. The color of the dot connected by dotted line shows p-value. The size of the dot shows an average percentage NR77. And the example data generates just one dot connected by dotted line. And each dot is generated in the same way. I tested germ-free mice as a negative control. T cells from germ-free mice failed to react most strain tested, which means the background noise of this experiment is very low. In sharp contrast, in a HCOM colonized condition, most of the strains in the community re-stimulated at least one type of T cell subsets, and many bugs stimulated multiple T cell subsets. It was surprising that one strain stimulates so many percentage of T cells. For example, Intestinobacter bartholletii stimulates 10% of P-Treg cells. As a result, the sum of all the T cells stimulated is far greater than 100%. This suggests the possibility that one T cell may react to multiple strains simultaneously. To understand this many-to-one strain TCR relationship, we examined TCR microbiome interaction at a higher resolution. We mixed and inoculated HCOM strains into germ-free mice. T cells were extracted and sent for single-cell RNA-seq and TCR-seq. Based on single-cell data, I chose 92 TCRs to make vectors of TCR genes. Even though 92 TCRs are a small pool of the entire TCR repertoire, we paid special attention to choose the TCRs that represent the whole repertoire. I established 92 different TCR hybridoma cell lines by this new high-throughput protocol. And then I did a co-culture experiment using one TCR hybridoma and one bacteria strain at a time using dendritic cells for antigen presentation to test TCR specificity. Supernatant was collected, and IL-2 was measured as a readout for TCR stimulation. This is the result of the co-culture experiment. Information about T cell subsets are shown left, and information about bacteria strains are shown top. GF6, the first T cell receptor, is selected as a negative control from germ-free mouse. This GF6 is one TCR, but shared by 4 T cells. 3 are P-Treg cells, and 1 is T-Treg cell. GF6 is not stimulated by any strain, meaning that this negative control is working well in this experiment. From now, we focus on this distinct part in the matrix, which clearly indicates one TCR interacts to many bacteria strains. 13 TCRs are a mixture of Treg cells and Th17 cells. These TCRs reacted to 19 Firmicutes strains. We hypothesized that there is a strongly immunogenic bacterial antigen that are shared across these 19 Firmicutes strains. To make a long story short, we performed genome-wide screening experiment and discovered that the T cell antigen is the substrate binding protein, which is a cell membrane lipoprotein that binds sugar substrate and uptakes nutrients into bacteria cells. It is surprising that this ordinary protein that takes up nutrients from outside of the bacteria cell is so strongly immunogenic. This protein is widely conserved across Firmicutes strains of bacteria. Our data shows even though gut T cell receptor repertoire is very diverse, these T cell receptors are targeting a small number of highly immunogenic bacterial antigen. And I believe that the substrate binding protein, the antigen that we discovered, is one of the most strongly immunogenic antigens from trillions of microbiome-derived molecules. Right side of the panel shows a structure prediction from AlphaFold 2. T cell epitope shown in red is in the conserved beta-barrel structure. Since 19 strains of Firmicutes strongly stimulated T cell receptor repertoire, we tested the hypothesis that deleting these 19 Firmicutes strains may create a therapeutic community. We colonized IL-10 knockout germ-free mice with HCOM or 19 Clostridia dropped out HCOM. We saw a much lower level of fecal lipocalin, which is the sensor of colonic inflammation in a dropout community compared to HCOM. This suggests that identifying T-cell inducing strains enable us to logically make a therapeutic community. Previous works showed that SFV induces SFV-specific Th17 cells. However, it has been unclear how a complex community of the gut microbiome interacts with the TCR repertoire. We found that a group of strains having a conserved antigen is recognized by a group of T cells from mixed T helper cell subsets. This may explain how a limited number of T cell receptors responds to highly diverse and dynamic commensal strains. Among 36 microbiome reactive T cell receptors, 13 recognize the substrate binding protein, so the targets of the gut T cell receptors are very limited to a few conserved bacterial antigens, and we believe substrate binding protein is one of the strongly immunogenic antigens in the gut. Even though we identified the antigen from mice, HCOM is selected from highly prevalent strains of the human gut microbiome, so it is possible that human gut T cells recognize the same antigen in our body. I'm excited to announce that from next month, I'm going to start my laboratory in the Department of Molecular and Cellular Biology at Harvard University. We aimed to understand and manipulate the T cell response to the microbiome and diet, and even beyond, including cancer and autoimmunity. Please let me know if one of you are interested in joining my lab. I would like to thank my postdoc supervisor, Michael Fishbach, and lab members and collaborators, and my funding. Thank you so much. Now it's over to James.
Yes, well, actually, first over to me, Kazuki. Many thanks
Yes.
to your fascinating presentation. So I'd like to tell you a little bit about our second speaker, Dr. James Moon.
Yes.
Dr. Moon is the John Gideon Searle Professor in the Department of Pharmaceutical Sciences, Biomedical Engineering, and Chemical Engineering at the University of Michigan, Ann Arbor. His interdisciplinary research program aims to develop novel therapeutics to improve the immune system in the setting of vaccines and immunotherapies. I will now hand the mic over to Dr. Moon.
All right, thank you very much for your kind introduction and the invitation to share our work here. So my lab at University of Michigan is working on novel ways to modulate the gut microbiome in the setting of cancer immunotherapy, as well as for potential treatment against autoimmune diseases. So in our work, we are approaching this slightly different from other laboratories. Rather than working on fecal microbiota transplant or selecting a collection of a bacterial consortium, we are working on prebiotics that can promote beneficial microbiome in animals and hopefully in humans as well. We screened many different FDA's generally recognized as safe ingredients in our studies and have identified a very potent prebiotic for changing the gut microbiome in a beneficial manner. That is inulin, a polysaccharide found in chicory root and Jerusalem artichoke. And this polysaccharide has a sweet flavor and has been used by food industry as a fat and sugar replacement. What we have found is that when we turn that into a gel form and dose orally in tumor-bearing mice, this has a very good impact on microbiome, resulting in good synergy with immune checkpoint blockers for eliminating tumor cells away. So I'll walk you briefly through that process. So in order to identify inulin, we performed these studies in tumor-bearing animals. We implant tumor cells away from the GI tract, and when tumors are established, we give oral treatments of different probiotics, as shown in these arrows. And we also give these immune checkpoint blockers. As you probably know, immune checkpoint blockers are monoclonal antibodies that activate exhausted T cells in the setting of cancer treatments. And as you know, a lot of patients are benefiting from immune checkpoint blocker treatments in the clinic, but not all patients are responding. So there's a much room for improvement for improving the efficacy as well as safety of immune checkpoint blockers. And in this study, we are trying to see if inulin, our select prebiotic, could synergize with these immune checkpoint blockers. And what the results are showing is the tumor size over time. When you have anti-PD-1 immune checkpoint blocker treatment alone, some mice are regressing tumors away. But interestingly, when you give native inulin orally, together with a systemic injection of anti-PD-1, you see more mice are eradicating tumors away. What's quite interesting is when you make gel form of inulin and dose orally, we see even more mice are eradicating tumors away in combination with anti-PD-1. And this results in a dramatic improvement in animal survival compared with anti-PD-1 alone. When you have inulin gel plus anti-PD-1, more mice are eradicating tumors away. And these mice are developing long-term immune responses because we can take the survivors and perform tumor cell rechallenge and show that 100% of survivors are resisting tumor engraftment, meaning that you can not only eliminate primary tumor, but also establish long-term memory response against the tumor relapse. We have shown this works in many different mouse models. This is black mice with a colon carcinoma model showing good synergy with inulin gel plus anti-PD-1 shown in red. We have shown this in melanoma model as well. And we have also done this in even a spontaneous colon cancer model where orthotopic colon cancers are formed. And in that aggressive setting, inulin gel treatments together with anti-PD-1 resulted in lowest tumor mass. So collectively, what these studies are showing is that in local GI cancer, as well as distant tumors, oral gavage with inulin gel could synergize with immune checkpoint blocker treatments. And I don't have much time to go into the exact mechanism of action, but what we have shown is that in tumor-bearing animals, this inulin gel goes into the colon tissue, resulting in microbial fermentation, resulting in release of microbial metabolites, short-chain fatty acids, that turn CD8 T cells into better anti-tumor memory like CD8 T cells. And when immune checkpoint blocker is given, these stem-like memory T cells are engaging tumor cells in peripheral tissues and eliminating tumors away. Switching gears a little bit, another challenge with the immune checkpoint blocker treatments is that there are side effects. In particular, non-specific activation of T cells could lead to colitis-like symptoms in the GI tract. And interestingly, over the course of our studies, we have found that inulin has a good impact on colitis. This is motivated by other prior studies showing that inulin consumption in humans is associated with a reduction in colitis and inflammatory bowel disease. So to test this idea, we recreated this situation in mice where we add a chemical DSS in drinking water to cause local inflammation and then give immune checkpoint blockers that results in colitis-like symptoms with a significant body weight loss, shortening of the colon length. And in this setting, oral gavage with inulin gel was able to protect mice against the colitis-like symptoms. So what this is showing is that inulin gel can not only improve efficacy of immune checkpoint blockers, but also reverse safety issues induced by immune checkpoint blockers. And based on this idea, we started to test more on how oral inulin gel treatments is leading to immune tolerance against inflammation in the GI tract. We believe gut microbiome changes induced by inulin gel is promoting a subset of T cells known as regulatory T cells. And the role of regulatory T cells in the GI tract is that they could induce tolerance to food allergens. For example, as you know, millions of people in the US and around the globe are allergic to many different food allergens. So we decided to test whether inulin gel together with food allergens could lead to tolerance. So we simply use the same inulin gel mixed with allergens. For example, egg white ovalbumin was mixed. And then when we dose that in allergic mice, what we do is we perform multiple oral immunotherapy. And then we do gastric challenge with the allergen itself to induce anaphylactic shock-like response. And what we have shown is that with inulin gel plus the allergen ova, those together, we see significant protection against body temperature loss, as well as anaphylactic-like symptoms, showing that inulin gel plus ova could protect mice against food allergy. Our approach is a platform approach because we can simply mix peanut extract onto the inulin gel and still show efficacy. This is another food allergy model with a cholera toxin with a peanut extract. With oral immunotherapy with the inulin gel, we can show significant protection against anaphylaxis induced by peanut extract challenge. And what's also what we have shown is that inulin gel alone has some impact. Peanut extract alone has some minor impact, but you really need both inulin gel plus the allergens together to have this beneficial impact. Mechanistically, what we are showing is that inulin gel is retained longer in the GI tract after oral dosing. In fact, if you look at native inulin, it goes into the GI tract and gets cleared from there within 4 to 8 hours of single dosing. But with our viscous inulin gel, after single dosing, they are retained up to 24 hours after single dosing. In particular, in small intestines, we see there are much more prolonged retention with inulin gel plus ova, as shown in this red. And what that does is that the inulin gel is allowing the local immune cells in the GI tract to take up the food allergens. This is showing macrophages like dendritic cells and tolerogenic disease are taking up ovalbumin delivered by inulin gel. And we believe that's how we are inducing regulatory T cells. In terms of gut microbiome changes, we have done extensive studies in small intestines as well as fecal microbiota. And what we have shown is that in mice treated with inulin gel plus ova, we see increases in beneficial microbes. These are microbes that are associated with the positive outcomes in food allergy patients. Agar LSA and Enteroraptus are going up with inulin gel treatments. And we also see reduction in pathogenic microbes associated with food allergy. In particular, Palonomas, Helicobacter, and Staph are all going down with inulin gel plus ova. And this results in significant changes in local immune responses. We see significant induction in FOXP3 positive regulatory T cells, as shown by the single cell RNA-seq in the small intestines. And we also see reduction in mast cells responsible for release of histamines. We also see reduction in allergen-specific IgE that drives the pathology in food allergy. Given that this is a dietary fiber-based approach, we can readily go to human trials. We have an ongoing healthy volunteer study where we have healthy volunteers taking up inulin powder or inulin gel over the course of 10 days. And we are analyzing their microbiome changes before and after oral consumption in hopes of finding whether the findings we have in mice will translate to human subjects. In particular, the initial readout shows that inulin gel is well tolerated without any major GI issues. We are asking people to take up to 20 grams of inulin gel per day. So we are worried that that may trigger some GI symptoms. But so far, we have very minimum GI symptoms. If anything, we see some gas and bloating and flatulence, which is indicative of microbiome-mediated fermentation of inulin. And we see they are as well tolerated as native inulin powder. So with that, we are aiming to go to human cancer patient trials later this year. As we have done in mouse studies, we will ask cancer patients who are undergoing immune checkpoint blocker treatments to consume our inulin gel, and we'll examine whether inulin gel can improve the efficacy of immune checkpoint as well as reducing the side effects. So with that, I want to thank you for your attention, all the lab members that have done the work, as well as our funding sources. Thank you.